Hi everyone, welcome to a new section, a new chapter. Today we are going to discuss our second chapter of biology textbook, that is biological classification. I hope you all have understood our first chapter, the living world. So this chapter is going to be a continuation of our first chapter. In our first chapter we discussed concepts like what is nomenclature, what is taxonomy, about classification, all those things, right? What is living. So in this chapter we are going to learn how classification is done. Okay. So the name of this chapter is biological classification. So let's start. Once again, myself Chitralekha, welcome to the new chapter. Okay, biological classification. So we are starting by looking into the history of classification. So we look who is the first person to do classification. So it is said that Aristotle was the first person to do classification. So he is known as the father of biology. It's an additional information. If you want, you can note it down. He is known as the father of biology. Father of biology, Aristotle did the first classification. So, how did he classify? He classified plants into trees, herbs and shrubs. Okay? Herbs, shrubs and trees. That was his classification. So, plants were classified into trees, herbs and shrubs. Okay, trees are the tallest plants. Then shrubs and then herbs. Okay? Yes. So, in basis of morphology. So, according to morphology, he classified plants into trees, herbs and shrubs. So, morphology means external features. So, this is according to height. Trees are the tallest, herbs are the smallest, shrubs are in between them. So, considering external structures, external feature which is the height, he classified plants into three. That is morphology. External features are called as morphology. Okay. So, plants were classified into trees, shrubs and herbs, and he classified animals into red-blooded animals and non-red-blooded animals. That means animals, those who have red-colored blood, and animals without red-colored blood. So, we humans are red-blooded animals. Okay. So, he classified plants into herbs, shrubs and trees, and he classified animals into red-blooded animals and non-red-blooded animals. Okay. Who is Aristotle? He is the father of biology. Okay. Yes. Then, next one is Carl Linnaeus or Carolus Linnaeus. Do you remember the name? Carl Linnaeus. He was the father of modern taxonomy. In taxonomy, we heard this name. He proposed binomial nomenclature. Yes, okay. So, Carl Linnaeus did two kingdom classification. So, his classification was known as two kingdom classification. He classified organisms into two kingdoms, that is kingdom Plantae and kingdom Animalia. Okay, his classification was two kingdom classification. He classified organisms into two kingdom Plantae and kingdom Animalia. Okay. Okay, so all plants were included in kingdom Plantae and all animals are included in kingdom Animalia. Is that clear? Okay. So, this classification had a lot of demerits. Why? Because there is only two kingdoms and we will see what were the demerits. First demerit, he did not distinguish between eukaryotes and prokaryotes. So, what is eukaryotes and prokaryotes? You have a lot of terms in this chapter. Okay, so that's very basic terms. Okay, eukaryotes. Okay, remember this. Eu means true. Eu means true. Okay, this is an important topic. Okay, so eukaryotes and prokaryotes. Karyo means nucleus. So eukaryotes means organisms with a definite nucleus. What is a definite nucleus? So, we know we have cells have nucleus. So, inside nucleus there is our genetic material. So, if this nucleus have a proper nuclear membrane, a proper nuclear envelope, we will call it a definite nucleus. So, in that case, the genetic material won't be naked. Okay. So, the cell will have a nucleus, inside it there will be genetic material and this nucleus will have proper nuclear membrane and nuclear envelope. In that case, we will call them eukaryote. That means organisms with definite nucleus is called eukaryotes. Okay, so what will be prokaryotes? Organisms without definite nucleus. So, in their case, their nucleus will be, their genetic material will be naked. Their cell won't have a proper nuclear envelope. So, their genetic material will be naked inside the cell. For example, in case of eukaryotes, there will be a nucleus inside which their genetic material is present. In case of prokaryotes, they won't have a nuclear membrane. So, their genetic material will be like this inside the cell itself. Okay. So, that is, he did not distinguish between eukaryotes and prokaryotes. So, he did not distinguish between organisms with proper nucleus and without proper nucleus. Okay. And unicellular and multicellular organisms. There is an example, eukaryotes and prokaryotes, bacteria and cyanobacteria. Bacteria and cyanobacteria lack a proper definite nucleus. So, they were included with organisms with a definite nucleus. So, that was a demerit. So, second point is he did not distinguish between unicellular organisms and multicellular organisms. So, what is unicellular organisms? Organisms which whole body is a single cell. That is unicellular organisms. And multicellular organisms, their whole body will be number of cells together forms their body, multicellular organisms. Okay, unicellular organisms and multicellular organisms. So, they were not differentiated, they were mixed up. Okay, example, Chlamydomonas and Spirogyra. Okay, though you will have to learn these terms, Chlamydomonas and Spirogyra. Chlamydomonas and Spirogyra was grouped with algae. So, algae is multicellular. So, unicellular Chlamydomonas and Spirogyra was grouped with algae. Okay. And he did not distinguish between heterotrophic fungi and autotrophic green plants. New terms, heterotrophic and autotrophic. What is heterotrophic? So, it refers to nutrition. Nutrients mean mode of nutrition. The way we get nutrients, the way we get energy. Okay, the mode. Heterotrophic means if an organism depends on another organism. If an organism depends on another organism for food, that is called as heterotrophic organism. Okay. If an organism can prepare food by itself, so it is independent, self-sufficient. So, it can prepare food by itself. That is called autotrophic organism. Okay. So, heterotrophic organisms are organisms that depends on another organism or other organisms for food, and autotrophic organisms are organisms that can prepare food by itself. So, heterotrophic organisms like fungi, you know fungi? Yes, fungi and autotrophic organisms like green plants. Green plants can prepare their own food by the method of photosynthesis. Right. So, this heterotrophic organisms and autotrophic organisms were mixed up. So, that was a demerit. Okay. Then, he did not distinguish between photosynthetic and non-photosynthetic organisms. Same, photosynthetic and non-photosynthetic organisms that can do photosynthesis and organisms that cannot do photosynthesis. So, he did not distinguish between these organisms also. So, that is also a demerit. And last one is, he did not distinguish between large number of organisms did not fall into either category. So, there is a number of organisms that does not fall into either categories. Okay. So, these were the demerits. So, first one is eukaryotes and prokaryotes. He did not distinguish between eukaryotes and prokaryotes. He did not distinguish between unicellular and multicellular organisms. He did not distinguish between autotrophs and heterotrophs, and photosynthetic and non-photosynthetic organisms. And at last, he did large number of organisms were did not fall into either category. So, there were organisms which cannot be grouped inside Plantae and Animalia. So, what about them? So, that was also a demerit. Okay. Yes. Okay. Next classification is R.H. Whittaker classification. Okay. R.H. Whittaker classified organisms into 5 kingdoms. So, his classification is known as 5 kingdom classification. This is the modern classification, like this classification is still followed. Okay. R.H. Whittaker's classification is known as 5 kingdom classification. So, in this classification, characters like cell structure, body organization, mode of nutrition, mode of reproduction and phylogenetic relationships were considered. Okay. What were the characters? Cell structure, how their cell is structured, about they have cell wall, in the organelles, like that. Cell structure of their cell. Then body organization, what makes their body, whether it is simply cells whether their body is made up of just cells or organs, tissues, like that, how their body is organized. Then mode of nutrition, how whether they are heterotrophic or autotrophic, like that. And then mode of reproduction, how reproduction is done, asexual, sexual, like that. And then phylogenetic relationships, evolutionary relationships. Okay, these were the characters that were considered while doing 5 kingdom classification. So 5 kingdom classification is done by R.H. Whittaker. So it is Whittaker's classification or 5 kingdom classification. Okay. So there are 5 kingdoms. Which are the kingdoms? First one, kingdom Monera. Next, kingdom Protista. Next, kingdom Fungi, kingdom Plantae and kingdom Animalia. Once again, kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae and kingdom Animalia. Very important, in this chapter we are going to discuss the first 3 kingdoms in detail. In next upcoming chapters you will discuss each of the kingdoms, like Plantae will be a chapter and Animalia will be another chapter. Okay, once again in this chapter we are discussing Monera, Protista and Fungi. Okay, so once again repeat with me, which are the kingdoms? Kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae and kingdom Animalia. Okay, so let's see the general. Okay, so let's see the merits of Whittaker's classification. Okay, what were the merits of Whittaker's classification? 5 kingdom classification. Fungi and plants were in separate kingdom. So in Linnaeus classification, fungi and plants were together. It was a demerit. So in Whittaker's classification, fungi and plants were in separate kingdoms. And prokaryotes were placed in kingdom Monera. All prokaryotes, all organisms without a definite nucleus were placed in a separate kingdom called kingdom Monera. And then all unicellular eukaryotes were placed in kingdom Protista. So there are eukaryotes like prokaryotes and eukaryotes. Except prokaryotes, all others are eukaryotes, in which there are multicellular eukaryotes and unicellular eukaryotes. So all unicellular organisms were given a separate kingdom called kingdom Protista. Okay, so these are the merits. Fungi were placed in a separate kingdom, prokaryotes were placed in a separate kingdom, kingdom Monera, and all unicellular eukaryotes were placed in a separate kingdom called kingdom Protista. Okay, okay. Now there is a system like 3 domain system. Here kingdom Monera is divided into 2 domains. So we will get 6 kingdoms. So just remember that there is a system called 3 domain system in which kingdom Monera is divided into 2, and now there is total 6 kingdoms. Okay, just remember that. Okay. Okay, so let's see the general characters of all these 5 kingdoms. Okay. So we will see the characters first and we will see how it differs in each kingdom. Okay. So first one is cell type, whether it is prokaryotic or eukaryotic. In kingdom Monera, what is the cell type of kingdom Monera? It's clear, it's prokaryotic, of course. Their nucleus does not have their genetic material is not covered with a nuclear envelope or nuclear membrane. So they are prokaryotic. They lack a definite nucleus. So kingdom Monera is prokaryotic. Then kingdom Protista, kingdom Fungi, Plantae, Animalia, all of them are eukaryotic. They have a proper nucleus. They have a definite nucleus. So they are eukaryotic. True nucleus. Okay, eukaryote. So U means true. Okay. Next is cell wall. Next character is cell wall. In case of Monera, Monera have cell wall. Does Monera have cell wall? Yes, Monerans have cell wall, and they are non-cellulosic. What is cellulose? Cellulose is a material by which cell wall is made. Commonly in plants, cell wall is made up of cellulose. In Monera, they have cell wall, but it is not made up of cellulose. So it is non-cellulosic. So cell wall is made up of polysaccharide and amino acids. In Monerans, their cell wall is made up of polysaccharides and amino acids. So it is non-cellulosic. It is made up of polysaccharides and amino acids. Okay. Next, in Protista, cell wall is present. In Protista, cell wall is present in some cell wall is present. In some protistans, cell wall is present, and if cell wall is present, they are cellulosic. Okay, cellulosic cell wall. In some protistans, cell wall is present, and these cell walls are cellulosic. Okay. Okay. In fungi, cell wall is present, but it is made up of chitin. Chitin. Remember that term. chitin. Cell wall is made up of chitin. So in fungi, cell wall is present which is made up of chitin. Then in plantae, cell wall is present which is made up of cellulose. Okay, so in plantae also cell wall is present. Cellulosic cell wall is present. The material by which cell wall is made, it is called cellulose. And in Animalia, we human beings comes under the kingdom Animalia. Do we have cell wall? No, we don't have cell wall. So for Animalia kingdom, cell wall is absent. Okay. Okay, so that's also clear. Cell type, cell wall. Now nuclear membrane and nuclear membrane. Yes, nuclear membrane. What about nuclear membrane? In Monera, nuclear membrane is absent. That's why they are prokaryotes. So in Monera, nuclear membrane is absent, and in all others, nuclear membrane is present. Okay. And then body organization. Body organization means with what their body is made. Is it just cells or tissues or organs, or organs together forms organ system? So what is the level of body organization? So in case of Monera and Protista, it is cellular. That means single cells represents their body. A cell alone forms a body. So they are cellular level. Monerans and Protistans have cellular body. And in fungi, which is multicellular or loose tissues, number of cells are formed, and number of cells together forms their body, or these cells are loosely bounded forming a loose tissue. Okay. In fungi, multicellular or loose tissues forms their body. In case of plants, tissues or organs forms their body. So cells together forms tissues, and these tissues together forms organs. That's how the body of plants are formed. Okay, in kingdom Plantae, it is their body organization is tissue level or organ level. Then Animalia, in animals, their body is made up of tissues. Tissues are present, and organs also forms their body, and organ systems also forms their body. What is organ systems? For example, our lungs is included in the respiratory system. Heart forms the part of circulatory system. Stomach, digestive system, like this. All these organs are part of a system. Okay, so tissues, organs, organ systems. Okay, this is their body organization. Then mode of nutrition. How, in which mode they get nutrients. So in case of Monerans, it's two types. It can be autotrophic or heterotrophic. Okay. So there is two ways in which they can get nutrition. Okay, that is autotrophic. They can produce it themselves, or heterotrophic. They depends on other organism. In autotrophic, there is two types: chemosynthetic and photosynthetic. In autotrophic, there is two types. That is chemosynthetic or photosynthetic. What is chemosynthetic? They can produce energy by chemical process. Using chemical process, if organism can produce food, they are called as chemosynthetic autotrophs. And if their production of food is by photosynthesis, the process of photosynthesis, they are photosynthetic autotrophs. Okay, that is chemosynthetic and photosynthetic. And in heterotrophs, there is saprophytic and parasitic. What is saprophytic? Saprophytic means an organism depends on dead or decayed organism for food. Dead and decayed organism for food. That is saprophytic. Parasitic, an organism will depend on another organism, another host, and harming the host. Results the harming of host. Okay, so mode of nutrition can be autotrophic or heterotrophic. In autotrophic, it can be photoautotrophic or photosynthetic or chemosynthetic. In heterotrophic, it can be saprophytic or parasitic. Is that clear? Okay. Then in Protista, their mode of nutrition can be autotrophic or heterotrophic. Again, autotrophic or heterotrophic. But in autotrophic, there is only photosynthetic. There is no chemosynthetic. Okay. In autotrophic, there is only photosynthetic. And in heterotrophic, okay, there is different types. Okay, so we'll just say heterotrophic. Okay, autotrophic and heterotrophic mode of nutrition. In fungi, heterotrophic. Yes. They are not autotrophic. They cannot prepare food by themselves. So fungi is heterotrophic, and the method is saprophytic or parasitic. Saprophytic means they depends on dead and decayed organisms for food, and parasitic depends on a host. Okay. And in plant A, they are autotrophic. Yes, plants are the primary producers. They can produce food by themselves. They don't have to depend on any other organism for food. So they are autotrophic. Their food production is by the process of photosynthesis. Okay, so the plants can produce food by themselves, hence they are autotrophic, and the process is photosynthesis. So they are autotrophic. Okay, and then animalia. Animalia, they are heterotrophic. Animals are heterotrophic. We animals, we can't produce food by ourselves. Like, we depend on other organisms for food. So we are heterotrophic. So our mode of nutrition can be holozoic or saprophytic, etc. There are different types. Holozoic means eating, ingesting food. We can ingest our food. That is holozoic. And saprophytic, depending on dead organisms. Okay, fine. So what were the mode of nutrition in Monera? Autotrophic or heterotrophic? In that, photo, chemo, and in heterotrophic there is saprophytic and parasitic. Protista, autotrophic, which is photosynthetic and heterotrophic. Algae, which is heterotrophic, and in which saprophytic and parasitic. Plants, it is autotrophic, which is by photosynthesis, and in animals it is heterotrophic, and we say holozoic and saprophytic. So this is the general characters of all this kingdom. It's the base of these kingdoms. Okay, is that clear? I hope it is clear. So yes, so we are going to explain the first kingdom in detail. We are going to see our first kingdom, Kingdom Monera. Okay, so are you ready for Kingdom Monera? Okay, let's start Kingdom Monera. So bacteria are the sole members of the Kingdom Monera. In Kingdom Monera, their sole members are bacteria. Okay, so this is a kingdom of bacteria. All bacteria are included in the Kingdom Monera. Okay, so bacteria are the sole members of Kingdom Monera. Okay, they are the most abundant microorganisms. We know in our world there is number of microorganisms. If you take some soil in your hand, there is thousands and billions of microorganisms are present. In this, most of them will be bacteria. So bacteria are the most abundant microorganisms in our world. Okay, they also live in extreme habitats. Extreme habitats means very harsh conditions. So it will be very hard for an organism to live in such conditions. So it is extreme conditions, harsh conditions. But bacteria can survive in such harsh conditions. Some bacteria can survive in such harsh conditions. They comes under this kingdom. So they also live in extreme habitats such as hot springs. Hot springs, yes, you know hot springs, the temperature will be high in hot springs. And deserts, yes, desert is a harsh condition. Snow, like below 0, the temperature is below 0 degrees Celsius. So in such a temperature also bacteria can exist. Even humans can't exist in such temperature. And then deep oceans, in deep oceans there is no light, pressure is less. So in such conditions also some bacteria can exist, where very few life forms can exist. So bacteria, these members of this kingdom, there are some members who can live in extremely harsh conditions. For example, hot springs and deserts, snow, and then yes, deep oceans, where other life form, the existence of other life forms are very hard. Okay, then okay, so Kingdom Monera, who are the members? Bacteria are the members. Okay, so let's see the first classification. On the basis of shape, bacteria are classified into 4 based on shape. First one is spherical shape. The spherical shaped bacteria is called as coccus. So spherical shape means like this. This is spherical shape. This is coccus. Okay. Okay, spherical shaped bacteria is coccus. Okay, spherical shaped bacteria is coccus. Then rod shaped bacteria. Rod shaped bacteria means like this. Okay. Rod shaped bacteria, they are called as bacillus. Once again, spherical bacteria, coccus. Rod shaped bacteria is called as bacillus. Is that clear? Yes. Then coma-shaped bacteria. Coma-shaped bacteria is called as Vibrio. So they will be coma-shaped. They will be like this. This is how coma is, right? Or like this. I think this is the right structure. Okay. Coma-shaped bacteria is called as Vibrio. Once again, spherical coccus, rod-shaped bacillus, coma-shaped Vibrio or Vibrium. Okay. Spiral-shaped bacteria is called as Spirillum. Spiral means like this. Okay. Spiral bacteria is Spirillum. Once again, spherical bacteria is coccus. If the shape of bacteria is spherical, it is coccus. If it is rod-shaped, it is bacillus. If it is coma-shaped, it is Vibrio. And if it's spiral-shaped, it is Spirillum. Once again, coccus, bacillus, Vibrio and Spirillum. Okay, is that clear? Okay, so that was classification based on shape. So next is bacteria as a group shows the most extensive metabolic diversity. So metabolic diversity means they can be autotrophs, they can be heterotrophs. So in such a way that shows a very diverse metabolism. Many types of metabolism can be shown by them. Some of the bacteria are autotrophic. So some are autotrophic. That means they can synthesize food by themselves. They does not depend on any other organism for food. Some can be autotrophic. Okay, yes, they can prepare their own food from inorganic substance, that is chemoautotrophic. Okay, we will discuss it. They may be photosynthetic autotrophic or chemosynthetic autotrophic. So some of the bacteria are autotrophic, they can prepare food by themselves. So it can be two types, photosynthetic or chemosynthetic. That means by process of photosynthesis or by process of chemical reaction, like from chemical reaction they will get energy. Okay, a vast majority of bacteria are heterotrophs and there is another majority which is heterotrophs. So majority of bacteria are heterotrophs. That means they depend on other organisms for food. Okay, yes, they depend on other organisms or on dead organic matter for food. Other organisms or dead organic matter, yes, anyway, other organism for food. So bacteria shows a very vast diversity of metabolism. It can be autotrophs or heterotrophs. In autotrophs, photosynthetic or chemosynthetic. Okay, next. Okay, so now we have two types of bacteria. Okay, so here, okay, first one we are going to discuss Archaebacteria. What is Archaebacteria? They live in some of the harsh habitats. So Archaebacteria is a type of bacteria who lives in very harsh conditions. Okay, some of them lives in very harsh conditions are included in this class, Archaebacteria. Okay. Example, extremely salty areas, very salty areas, which is called halophiles. So bacteria that can exist in extremely very salty conditions are called as halophiles. Okay, halophiles. Then hot springs, that means high temperature areas, conditions in which temperature is very high. So they are known as thermoacidophiles. Okay, bacteria which lives in very salty conditions are known as halophiles, and those who lives in hot springs are known as thermoacidophiles. And then those who lives in marshy areas, they are called as methanogens. So three type of bacteria: extremely salty areas, halophiles; hot springs, thermoacidophiles; and marshy areas, methanogens. So these are examples for bacteria that lives in very harsh conditions. These names are important: halophiles, thermoacidophiles and methanogens. Terms are very important. Okay, yes. They have a different cell wall structure that helps them to survive in extreme conditions. So it is their cell wall. It is their cell wall that protects them in such harsh condition. So without damaging their genetic material, their cell is kept intact by their cell wall. So this cell wall protects them from this harsh condition. Okay, so cell wall is very important. Okay, and there is a case where methanogens, methanogens are present in gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane, biogas from the dung of these animals. You know, there is biogas production. If there is a cattle farm near your house or somewhere, you may have seen there is biogas production. From the dung of these animals, biogas or methane is produced. So this is due to the presence of methanogens in the gut of ruminant animals. What is ruminant animals? Grazing animals. Okay, herbivores, grazing animals are called as ruminant animals. So in the gut of several ruminant animals, methanogens are present. Okay, methanogens are present. So they are responsible for production of methane or biogas from the dung, from the excreta of these animals. Okay, is that clear? It's an example. Methanogens are a classic example. Okay, yes. So Ary bacteria, some lives in harsh conditions, some examples, and their cell wall is important. And then what is methanogens? Importance of methanogens. So next type of bacteria is eubacteria. Once again, eu. What is eu? Eu means true bacteria or eubacteria. Okay, let's see what is eubacteria. Eubacteria or true bacteria. Eu means true. Okay. Presence of a rigid cell wall and if motile a flagellum. So they have a rigid cell wall. Yes, eubacteria has a rigid cell wall, and if they are motile, they will have a flagella. What is flagella? Flagella is a structure that helps for movement in or locomotion in bacteria. It will be, this is flagella. Consider this as bacteria. For movement, it will have a special structure called flagella. So this is flagella of a bacteria. So if motile, they will have a flagella. So if they are motile, if they can do movement, it will be with the help of a structure called flagella. Okay. This group has photosynthetic autotrophs. Yes. Chemosynthetic autotrophs and heterotrophs. So in this group of Ary bacteria, there are photosynthetic autotrophs, chemosynthetic autotrophs and heterotrophs. Okay. Okay. For example, for photosynthetic autotrophs, bacteria, they can produce food by itself with the process of photosynthesis. Example is cyanobacteria. It's important. Cyanobacteria is a photosynthetic autotroph. Okay. So photosynthetic autotroph, example cyanobacteria. Okay, now we will discuss cyanobacteria. What is cyanobacteria? Who is cyanobacteria? Cyanobacteria is also known as blue-green algae. Okay, blue-green algae. It is called as blue-green algae. This is a new name given for it. Blue-green algae. Name is important. It's often asked. They are unicellular, colonial or filamentous freshwater or marine or terrestrial algae. Okay, so they are unicellular, colonial or filamentous, freshwater or marine or terrestrial. That means they are unicellular. They are made up of a single cell. Yes, bacteria are made up of a single cell. But what is colonial? They live as colonies. That means number of individuals lives together like this. In a region, number of them lives together, and there will be a division of labor. Okay, there will be a division of labor in this colony, living together as a group, as a colony, single individuals living together as a group. Okay, that is colonial, living as a colony. They live as a group in an area. So they don't live freely, they live as a colony, number of them together. Okay. Or filamentous, they forms a filament like. Okay. And freshwater or marine, they can be seen in freshwater or marine. What is freshwater? Freshwater, example, rivers. Rivers are freshwater. So those who are present in river like freshwater, they are freshwater. And marine, example, sea, ocean, that's marine. So they can be seen in freshwater or marine. And terrestrial, even in terrestrial habitat. Okay, in land also. So they are unicellular, colonial, filamentous or freshwater or marine or terrestrial. Okay. They have chlorophyll A like green plants, hence the name blue-green algae. Blue-green algae. So they have chlorophyll A in them, so like green plants. So that helps them for photosynthesis. They are photoautotrophs, right? So this chlorophyll A helps them to do photosynthesis. So like green plants, they also possess chlorophyll A. Okay. Okay. The colonies are generally surrounded by gelatinous sheath. So in this colony, there will be a gelatinous sheath. Covering this colony, there will be a gelatinous sheath. Okay, this is a colony. Individuals are present together, are grouped together. Okay, so there will be a gelatinous sheath around their colony. Their colonies are generally surrounded by a gelatinous sheath. They often form blooms in polluted water bodies. So if there is a water body and it is polluted, they will form a bloom. Bloom is, many of them together will form a special structure called bloom. This blooms can produce toxins that is harmful for organisms in that water body. So, they often forms blooms in polluted water bodies. Some of them can fix atmospheric nitrogen in specialized cells. So, what is fixing atmospheric nitrogen? So, there is nitrogen in our atmosphere. In our atmosphere there is nitrogen. In case of plants, plants need nitrogen inside their body. They can't directly absorb nitrogen from the atmosphere. So, they need to get nitrogen through the soil. So, this process is done by these bacteria. So, they will convert atmospheric nitrogen into compounds which can be absorbed by plants. So, some of them can fix atmospheric nitrogen in their specialized cell. Some bacteria will have a special cells. They can convert the nitrogen in the atmosphere into absorbable forms for plants. Okay. Some of them can fix, that is called fixing atmospheric nitrogen, okay, in specialized cells called heterocyst. So, heterocyst is the cell that helps in fixing atmospheric nitrogen. So, some of them can fix atmospheric nitrogen with their in specialized cell which is called as heterocyst. So, here 2 examples are given, which is nostoc and anabena, okay. I hope you can see that. Okay, nostoc and anabena. Okay, these are the example for nitrogen fixing bacteria. They are nostoc and anabena. Once again, we were talking about cyanobacteria, which is called blue-green algae. They are unicellular, colonial or filamentous, freshwater or marine, and terrestrial or maybe terrestrial also. And they have chlorophyll A like green plants. That's important. Hence they are called as blue-green algae. Hence that's why they can do photosynthesis. Then they form blooms in polluted water bodies, and then some of them can fix atmospheric nitrogen in their cell called heterocyst. Heterocyst is also an example. Nostoc and anabena. Here I have given a picture. This is nostoc. Okay, this is a nostoc, and this is heterocyst. Nitrogen fixing specialized cells. This is heterocyst, and this is a mucilage sheath. We already said there is a gelatinous sheath around the colony. So that is a mucilaginous sheath. Okay, is that picture clear? It's from your NCERT textbook. Okay. Okay, so that's also completed. Next is chemosynthetic autotrophic bacteria. So yeah, last cyanobacteria was autotrophic, like they were photosynthetic autotrophic bacteria. They are autotrophic, and by the process of photosynthesis they produce food. This is chemosynthetic autotrophs. So they can produce food by themselves with the process of by chemical reactions they will produce food. So let's see how. They oxidize various inorganic substances such as nitrates and ammonia. So there are inorganic substances like nitrates and ammonia. So they will oxidize these inorganic substances. So these inorganic substances undergo the process of oxidation. The released energy is used for their ATP production. So by this process of oxidation, the energy an energy will be produced. By the process of by oxidation of this inorganic substances, an energy will be produced. This energy is used for ATP production. Okay, this is how they produce energy. Okay, they play a great role in recycling nutrients like nitrogen, phosphorus, iron and sulfur. So recycling, the process of recycling nutrients are done by them. So they can recycle these inorganic substances like nitrogen, phosphorus, sulfur, all of them. Okay. Okay, I hope it's clear. They oxidize inorganic substances and energy is released. Using this energy, they produce ATP, and they play a great role in recycling nutrients like nitrogen, phosphorus and iron and sulfur. Okay, is that clear? Okay. So next is heterotrophic bacteria. We have discussed autotrophic bacteria, chemosynthetic or photosynthetic, both. Okay. Now next is heterotrophic bacteria. Most abundant bacteria. So in the group of Monera, most of them are heterotrophic. That means most of them depends on other organisms for energy or for food. Okay. So most abundant, heterotrophic bacteria is the most abundant bacteria. Majority are important decomposers. Majority of these bacteria are decomposers. What is decomposers? they can decompose different substances. In soil, they can decompose. Okay, so majority are important decomposers. They are helpful in making curd from milk. So these are some applications. So applications. Remember, they are helpful in making curd from milk. From milk, they can we do that in our house itself. Okay, in our home. What we produce curd from milk. Okay, you know how it is done, I hope you do. So they are helpful in making curd from milk. That is done by this bacteria, okay, heterotrophic bacteria. And production of antibiotics, yes, it is using this heterotrophic bacteria, antibiotics are produced. So these are applications, making curd from milk, production of antibiotics, fixing nitrogen. In legume roots, okay, in legume roots, nitrogen fixing is done by this heterotrophic bacteria, okay. These applications are important. Curd from milk, production of antibiotics, fixing nitrogen in leguminous plants, okay, legume roots, okay. Some are pathogens, pathogens causing damage to human beings, crops, farm animals and pests. What is pathogens? The disease causing organisms, disease causing bacteria are known as pathogens. So some of them are pathogens. They cause disease or they cause damage to human beings. They cause disease to human beings. There are a lot of bacterial diseases. Okay. In crops, yes, plants are also affected by bacteria. And farm animals, yes, animals, pets, all of them are affected by bacteria. There is number of disease caused by bacteria. So some of them are pathogens. Then there is an example for some of bacterial diseases, that is cholera, typhoid, tetanus, citrus canker, and all these well-known diseases are caused by different bacteria. So yes, cholera, typhoid, tetanus, citrus canker, these are some diseases caused by bacteria. Okay, is that clear? A citrus canker is a plant, is a disease, disease in plants, plant disease, and all these are this animal disease, cholera, typhoid, tetanus, citrus canker. These are well-known disease caused by bacteria. Is this portion clear? I hope it is. Okay, so our last, what is it? Reproduction in bacteria. How reproduction happens in kingdom Monera. Let's see. Mainly by fission. What is fission? Fission. One cell dividing into two cell or two or more cell, okay, that is fission. This is binary fission, two cell, okay, binary fission. So a cell giving rise to more cells, like, yeah, it is called fission. Okay, you know, do you remember this image? And then it become two cells. Okay, simply one cell giving rise to more than one cell is fission. Okay, so reproduction is done mainly by fission and under unfavorable conditions. So this fission takes place in favorable conditions. So what is favorable conditions? For example, for bacteria, 37 degrees Celsius, which is our body temperature, is the most favorable temperature. Okay, so under favorable conditions, they will reproduce. Reproduction is done by fission. Under unfavorable conditions, when the condition is not favorable, even like the conditions are unfavorable, they will produce by spores. Spores are specialized structures that helps in reproduction. So under unfavorable condition, they will produce spores, or they also reproduce by a sort of sexual reproduction, like sexual reproduction. It is not exactly sexual reproduction, but it is a type of sexual reproduction, a sort of sexual reproduction. It is DNA transfer from one bacteria to another. That means DNA, genetic material, is transferred from one bacteria to another. Okay. Okay. So yes. So reproduction mainly it is by binary fission. Under unfavorable condition, it can be by using spores. Spores are specialized structures, or by a sort of sexual reproduction, which is DNA transfer from one bacteria to another. From one bacteria, their genetic material is transferred to another bacteria. That is also a kind of reproduction. So next one is, this is our last part of kingdom Monera, mycoplasma. Okay, what is mycoplasma? Completely lacks cell wall. Mycoplasma are the smallest living organisms. Okay, they are the smallest living organisms. It is written here. They are the smallest living cells ever known. Smallest living cell. So that's a general question. So the smallest cell ever known is mycoplasma. They completely lack cell wall. They don't have a cell wall. So it will be just a cytoplasm and some genetic material. That's it. Okay. So they does not have a cell wall. They are the smallest living organ or living cells. Okay. They can survive without oxygen. Yes. There are organisms that can survive without oxygen. So these organisms can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. So they can cause disease also. Okay. So they are the smallest living cells ever known, and they lack cell wall, completely lack cell wall. They can live without oxygen, and many mycoplasma are pathogenic in animals. Okay, animals and plants. Is that clear? Once again, we discussed kingdom Monera. So what were the topics we discussed? Generally, we said some points, and then we spoke about classification based on shape, that's important, spherical coccus, rod shaped bacillus, coma shaped vibrio, and spiralum. And then some more points. There are two types of photoautotropic and chemoautotropic. Then we discussed Archaebacteria. Archaebacteria, they are primitive, they are halophiles, thermoacidophiles, methanogens, important, and this also methane production. And then Eubacteria, it is known as true bacteria, in which we discussed cyanobacteria and its properties, heterocyst, nostoc and anabina, and some points also. And then chemoautotropic bacteria. What is chemoautotropic bacteria? They can produce energy by chemical reaction. Yes. And then heterotropic bacteria, most abundant, decomposes, some application points, and then these diseases, and then last their reproduction, which is done by fission, spores, and DNA transfer also. Okay. Then mycoplasma, these points also. It is the smallest living cell ever known. Is that clear? So here we complete our first session, which is I gave the general introduction and general characters of all kingdoms also, and then we discussed kingdom Monera. So in next sessions we will discuss kingdom Protista, kingdom Fungi, and in general kingdom Plantae and Animalia. Okay, that's it. So thank you. I hope you all understood the whole topic or the whole session. Thank you.